Hey everyone, let's dive into our second chapter looking at gluconeogenesis. Gluconeogenesis is the making of new glucose. So it is the reverse of what we just went through in glycolysis, which was breaking down glucose. Gluconeogenesis is an anabolic pathway. So one of few anabolic pathways we'll be covering at the beginning of these topics. Remember, right now we're generally focusing on catabolic or catabolic pathways, or the breakdown of different biomolecules. So if you look, here is kind of a diagram of all of, not all of, but many of the pathways that we will be going through that are involved in energy generation. And you can see here for the making of sugars, for when sugar is needed, for example, if glycogen needs to be built back up, or for example, in plants, when starch or sucrose is being made inside a plant. The only way to actually make these sugars is backwards through, partly at least through gluconeogenesis. Mammals can't convert fats into sugars. So there needs to be some type of pathway for getting glucose from any other precursor that isn't already a sugar, to then be used for building sugars, or if you want to convert some other molecule into sugar so that you can then break it down for energy. So gluconeogenesis is used kind of across the board by animals, plants, fungi, and microorganisms for different reasons. It's one of several mechanisms that humans use and other animals use to maintain their blood glucose levels. This is to help avoid what's called hypoglycemia, or low blood sugar. Our circulating levels of glucose are very important because glucose not only is used as energy, but it is used to signal to other cells and tissues in our body whether we are low or high on energy in general. So it's a signaling molecule, and it is also an actual energy molecule. So we want to avoid having low circulating levels of glucose, because that would signal to our body that we are low on sugar. So one way to show that we are not low on sugar is to increase the levels of circulating glucose by using gluconeogenesis. Of course, if we were low on sugar, if our glucose levels were low, you would also be using potentially this gluconeogenesis. Other means include the degradation of glycogen and fatty acid catabolism. So these are other reasons that you would be using gluconeogenesis. So in our last chapter in glycolysis, we talked about general fates of glucose and different pathways it could go into. One of them, of course, was through glycolysis and generating energy. Today, we're going to focus on kind of two pathways in this glucose fate plot, which is the reverse of glycolysis. So going from pyruvate back to glucose, gluconeogenesis. And also we'll be talking about the pentose phosphate pathway, which is another fate of glucose in which you can either generate this ribose 5-phosphate, or again, go backwards to generate glucose. So two different interconnected fates we'll be talking about. First, let's look at the direct pathway of gluconeogenesis versus our pathway of glycolysis. Things to note is that glycolysis is going to occur mainly in the muscle and the brain. These are areas in which you will need rapid breakdown of sugar to produce energy, usually. So, brain and muscle cells are very hungry and require energy. Gluconeogenesis is going to occur mainly instead in the liver and the kidney. These are areas in which you potentially are taking other biomolecules, for example, in the kidney, other amino acids, et cetera, to build back up sugars, or in the liver, where we also, again, are recycling molecules and building up stores of sugar. These pathways are opposing, but both thermodynamically favorable. So either direction is thermodynamically favorable. And of course, the end product of one is the starting product of the other. So they are complete opposites of each other in that sense. They, however, do have some differences. There are irreversible steps in glycolysis. If you remember, we focused on three particular enzymes: hexokinase, phosphofructokinase 1, and pyruvate kinase. These are irreversible steps. They are steps associated with high energy. And these steps, to bypass them in gluconeogenesis, are different. So you'll see all of the reversible steps of glycolysis act in gluconeogenesis. They are, in fact, reversible. They can go in either direction. However, these three enzymes are different. They have different enzymes and different pathways in gluconeogenesis, and they are regulated differently. So these are the only steps that differentiate glycolysis versus gluconeogenesis. Otherwise, they're using the same mechanisms. So let's take a closer look at these steps. In gluconeogenesis, the three irreversible steps are, again, a whole nother set of enzymes that are used for gluconeogenesis versus glycolysis. We'll look down here at the bottom at the first set of enzymes. You can see that our very last step in glycolysis, which uses pyruvate kinase, is instead reversed through a series of two steps. So this is one of the first major differences in gluconeogenesis, is that you're doing this two-step process, and you can see that we instead use the enzymes pyruvate carboxylase and PEP, or this is phosphoenolpyruvate, PEP, carboxykinase. So through these two steps, we reverse pyruvate kinase, and we are able to take pyruvate and make phosphoenolpyruvate. Again, up here in gluconeogenesis, we're using fructose 1,6-biphosphatase 1 instead of our very important enzyme from glycolysis, phosphofructokinase 1. And lastly, the first step in glycolysis, which is the last step in gluconeogenesis, is going to use this enzyme glucose 6-phosphatase instead of hexokinase. Let's take a closer look at that series of enzymes at the beginning of gluconeogenesis. These are steps 1 and 2 of gluconeogenesis. And remember, they're the reverse of the end of glycolysis. So here we're looking at taking pyruvate and eventually turning it into phosphoenolpyruvate. These this again happens through a series of 2 steps, which is one of the major differences between glycolysis and gluconeogenesis. And they are very energy consuming. This step in particular, going reverse of pyruvate for gluconeogenesis, is the energy consuming portion of the whole phase of gluconeogenesis. It requires also the addition of another molecule, which is bicarbonate. So with addition of bicarbonate and pyruvate, we go through the first step, which is pyruvate carboxylase. This converts pyruvate into an important molecule called oxaloacetate. Oxaloacetate we will see again when we talk about the citric acid cycle because it is one of the key molecules in the citric acid cycle. So oxaloacetate is a circulating molecule, a circulating amino acid that we will see numerous times again. An interesting thing is that this step, taking pyruvate and making oxaloacetate through pyruvate carboxylase, happens in the mitochondria. So if you recall from glycolysis, glycolysis occurs in the cytosol. So to start the reverse of glycolysis, to start through gluconeogenesis, we actually have to get pyruvate out of the mitochondria and first turn it into oxaloacetate. This step is stimulated by high levels of acetyl-CoA, and it's inhibited by high levels of ADP and glucose. So this again makes sense because if you have high ADP, that means you are low on energy. That means you do not want to be spending energy to build up glucose, right? You actually want to be breaking down glucose. Also, if you have high levels of circulating glucose, you do not need to start building up glucose again. So this is an example of when one of the products of a reactant inhibits the beginning step, the beginning stages. Now, this enzyme uses a cofactor called biotin, and we will see this cofactor again. Biotin helps to carry carbon dioxide. It's going to use that source of carbon dioxide, which, if you remember, is bicarbonate. Notice that we're using this CO2 on bicarbonate. Bicarbonate comes from our blood. Remember from our blood CO2 cycling, our blood buffering, we turn CO2 in our blood into bicarbonate. So the bicarbonate is our source of CO2, and biotin, which is part of the enzyme pyruvate carboxylase, actually holds on and carries that CO2 from the bicarbonate to transfer it onto pyruvate. So you can see in this very simplistic diagram, we have our enzyme pyruvate carboxylase. This is a very simplified view of it. So this is like our binding site. It's a close-up view of pyruvate carboxylase, the whole enzyme. And within the inner portion of the enzyme, there is a lysine amino acid, which is attached to this molecule called biotin. So biotin acts as a cofactor. Remember, that's a molecule that's incorporated into an enzyme to help it function. And it is going to carry the CO2. It's going to bind the CO2 on site 1. And it actually is very pliable because it's long and has this long chain. And it moves to site 2, where it is then going to link onto the bound pyruvate, turning pyruvate into oxaloacetate. Notice that pyruvate is a 3-carbon long molecule and oxaloacetate is a 4-carbon long molecule. The second step in this is going to then be our generation of phosphoenolpyruvate from the oxaloacetate we just made. Or again, to PEP. Remember, this is abbreviated as PEP, phosphoenolpyruvate. So this is interesting because the CO2 that we just put onto our pyruvate to make oxaloacetate is actually now going to be removed, and it's going to be removed with the help of a GTP. This is a guanosine triphosphate, very similar in action to ATP, adenosine triphosphate. And instead of an adenosine nucleotide, has a guanosine nucleotide on the end of it. But its function is the same in that it uses those phosphate linkages, it breaks them to generate energy. So the phosphate on our phosphoenolpyruvate comes from a GTP in our gluconeogenesis. So instead of ATP, we use GTP in the gluconeogenesis. During this process, we also are removing that CO2 from the oxaloacetate that we just put on, resulting in our phosphoenolpyruvate. Now, this whole action, converting oxaloacetate into phosphoenolpyruvate, can occur in the mitochondria or it can occur in the cytosol. This is depending on an organism. In humans, we can do this in the cytosol. So remember again that the first step of making oxaloacetate only occurs in the mitochondria. And then this step occurs in the cytosol. And again, all of the other steps of our glycolysis and the other steps of gluconeogenesis, all of those occur in the cytosol. So we have this interesting moment where our pyruvate that we need to actually make glucose, if we're doing gluconeogenesis, has to come from the mitochondria. This is part of the pyruvate malate cycle. This helps regulate the levels of pyruvate that are in the cytosol for use in energy generation or to build up our glucose. Now, the mitochondria itself is permeable to select molecules. It can let out malate, it can let out, it can go transfer in between. Malate, phosphoenolpyruvate, or PEP, right? Also pyruvate can go in and out, but oxaloacetate can't escape. So pyruvate, even though it can go in and out, it needs to first be transformed into oxaloacetate to start our gluconeogenesis, which cannot escape the mitochondria. So we have an issue here. Our oxaloacetate eventually has to come out into the cytosol to continue the gluconeogenesis, right? To continue step 2 using our GTP and making PEP, phosphoenolpyruvate. But to do this, it has to first be converted into malate. So oxaloacetate and malate can be converted between each other within the mitochondria. And through this, the malate can then be shuttled out of the mitochondria, converted back into oxaloacetate. So you have a back and forth here. And then that oxaloacetate can continue going through these steps, making the PEP, which then continues through the rest of our gluconeogenesis. Now, to do this conversion of oxaloacetate to malate, we use an enzyme called malate dehydrogenase. And as shown here, this malate dehydrogenase has to exist in a form in the mitochondria and also in the cytosol, because one form is used to convert oxaloacetate to malate inside, And then another form is used to convert malate back into oxaloacetate on the outside in the cytosol. So this is a case where we have an enzyme that has two forms, a mitochondrial form and a cytosol form, to help with this function of moving oxaloacetate between the mitochondria and the cytosol. So again, our additional enzymes that are important to the gluconeogenesis after we generate PEP are going to be fructose 1,6 biphosphatase, phosphatase 1, which is going to take fructose 1,6 biphosphate and make it into fructose 6 phosphate. This is going to not generate ATP as its opposite does. It instead is taking water and an inorganic phosphate to add on to cleave off of that fructose 1,6 bisphosphatase and generate the fructose 6 phosphate. So even though there is a cleaving of a phosphate here, it does not generate ATP. So this is not an energy generation step, even though the reverse uses ATP. Similarly, glucose 6 phosphate to glucose, that last step in gluconeogenesis or the first step in glycolysis, in reverse also does not generate ATP. So even though the reverse uses an ATP to make an ADP in glycolysis, going this way through gluconeogenesis doesn't do the opposite. It does not just make an ATP. So important steps here, we're going to be using different enzymes. They don't act in the same function. And those steps that were using energy in glycolysis do not make energy in gluconeogenesis. So let's talk about a relation of gluconeogenesis to disease and human health. Remember that the control of gluconeogenesis is going to be mediated by this peptide called glucagon. Glucagon is a hormone peptide. Its inverse hormone peptide is insulin. And glucagon is released when blood glucose is low. So insulin is released when blood glucose is high to signal that we have too much blood sugar and the glucose needs to be broken down. Glucagon is released when blood glucose is low, and it helps signal that we need to be generating glucose. People who have type 2 diabetes, this is the type that you are more familiar with. This is diabetes that is induced by diet and other environmental factors. These people, it's usually marked by an excess of glucagon and insulin resistance. So we usually have excess glucagon and insulin resistance in the body. And this leads to increased levels of hyperglycemia. So one of the ways to reverse this, one of the drugs that is used to counteract part of type 2 diabetes is a drug called metformin. This is going to help reduce blood glucose just by inhibiting gluconeogenesis. So it targets one of the problems. So we have this kind of dual problem of we have too much glucagon around. So that keeps telling our body, make more glucose, make more glucose. And we have insulin resistance, meaning we're not listening to the insulin that is saying, no, you need to break down glucose. So targeting half of this is to essentially inhibit or reduce the gluconeogenesis by stopping the action of glucagon. So it's targeting the glucagon side of this. You can see here, this is actually a diagram showing the full cellular signaling of metformin and how it stepwise shows the signaling reaction that then shuts down our gluconeogenesis. The main thing here is that it's going to signal to an enzyme called AMPK, which you can see inside the cell, which inhibits an enzyme we just talked about, phosphophenylpyruvate carboxykinase, and it inhibits glucose 6 phosphatase. These are two enzymes that are part of the pathway for gluconeogenesis. So by inhibiting these enzymes, the body then cannot go through the process of converting pyruvate back into glucose because the enzymes it needs for that pathway are now inhibited. So this is one way metformin is often given in tandem with taking insulin for people who are diabetic because it helps to slow the amount of blood sugar that is built up in the blood by stopping any gluconeogenesis that would be occurring. So let's talk about the end product of gluconeogenesis. We read out our formula for glycolysis, but let's look at the total formula and energy spent using gluconeogenesis. So again, remember, we're going to be going the reverse, pyruvate to glucose here. This is in total going to be taking 2 pyruvate. It's going to use 4 ATP. These are the reverse of the 4 ATP that we normally would be seeing in glycolysis. So our steps in which we are generating, we are now going to be using those 4 ATP. It also has to use 2 GTP. Remember that these steps in gluconeogenesis that take our pyruvate and make it back into PEP require a GTP per pyruvate, right? So for 2 pyruvates, we need 2 GTPs. Remember also in our reverse steps that in glycolysis, we actually were going to generate 2 NADH. So in gluconeogenesis, we use 2 NADH and convert it into 2 NAD. Of course, also our H plus and water is utilized as well in the formation or in the usage of ATP and GTP. So you can see to do gluconeogenesis, it is very expensive. It costs 4 ATP, 2 GTP, and 2 NADH. Remember that glycolysis, its net generation of ATP is only 2 ATP that it makes, and it makes 2 NADH. So going in reverse to make glucose is very expensive. Glucose is a reserve of energy, so it makes sense that to take something that is not as energetically favorable, a small pyruvate, and try to convert it back into glucose, which is an energy reserve, is very expensive. So gluconeogenesis is only really done when it is physiologically necessary in humans. So in cases in the brain, nervous system, or red blood cells, these are going to generate ATP only from glucose. These areas thus are going to need glucose And in cases where they are completely depleted of glucose, for example, starvation, vigorous exercise, they can't get ATP from any other source. So, again, brain, nervous system, red blood cells, these rely on glucose for their energy. So, these particular areas during times of stress and starvation and exercise are going to then put in the energy to make glucose. So they will try to make glucose from other precursors, amino acids, not fats, amino acids and other things, so that they can then use that glucose to get energy. Animals can produce glucose from sugars or they can produce it from proteins. So they can take things like pyruvate, which we just talked about. If you have excess pyruvate, they can take lactate if they made lactic acid, or they can take oxaloacetate. And they can also take amino acids from various intermediary steps to then build up glucose. But they cannot produce glucose from fatty acids. This is a key point. Um, the product of fats is instead acetyl CoA, and there is no ability, they cannot have a net conversion of acetyl CoA to oxaloacetate. So, oxaloacetate on its own can be used, but we cannot do this conversion of fatty acids to oxaloacetate to be used during gluconeogenesis. Now, other cells, plants, yeasts, and other bacteria actually can take fats and then turn it directly into glucose. So again, some of the main precursors for gluconeogenesis are going to be lactate, which I just talked about. It can be in the form of lactate or pyruvate, depending on how it's been converted. This is going to be produced from exercising in muscles and red blood cells and can be reconverted into pyruvate in the liver. The liver has the ability to do this, and then that pyruvate can then be built back up into glucose through gluconeogenesis. One way that uh, you can get a precursor to gluconeogenesis that's not actually directly from fats is through glycerol. Glycerol often comes from triacylglycerols, which is a part of fatty storage of fats, right? It's the backbone to those fats. If you remember, we have our glycerol that then has our fatty acid chains. So the actual fatty acid chains on our triacylglycerol cannot be converted into, in humans, cannot be converted into glucose. But this backbone piece, this little backbone piece actually can be, that glycerol backbone can be converted into dihydroxyacetone phosphate, which is part of the glycolysis and gluconeogenesis pathway. So it can feed in right there and then be used to generate glucose through gluconeogenesis. And of course, one of the other major ways to generate glucose if we're have low blood sugar is through amino acids. These can come from a various amount, a variety of amino acids from the citric acid cycle. They can be already converted back into pyruvate through other means. And then, of course, that feeds back up into gluconeogenesis. This is going to be the major source of glucose during extended fasting. So when you are very, very, very low on sugar and low on energy, you start to use protein to make amino acids, right? You break down protein, make amino acids, and then those amino acids can be used to make glucose for energy. This is why people during extreme fasting actually start to lose some of their muscle mass because we're breaking down our proteins and our muscles to help feed into making glucose. The amino acids that can be turned into glucose have a name. They are called glycogenic amino acids. These are almost all of the amino acids. In fact, if you look, only leucine and lysine are unable to furnish carbon or give carbon to make glucose synthesis. And these amino acids can come from a variety of areas. They can feed into the citric acid cycle, which we'll be going into more. So they can feed into various points of our metabolism pathway and generally eventually be turned into pyruvate, of course, if not directly, then through a series of steps, which then feeds back into gluconeogenesis. Now let's talk about another pathway which has its fate tied to glucose. It is called the pentose phosphate pathway, or the PPP. The main products of the pentose phosphate pathway is to produce another electron carrying molecule called NADPH, not to be confused with its close cousin, NADH. NADPH has a slightly different structure, though its role is similar to NADH. It also generates ribose 5 phosphate, which should be familiar if you remember looking at our structures of nucleotides. Ribose is a structure that helps build up our nucleotide precursors in RNA, and also deoxyribose can, of course, be used in DNA. Now, the reverse of this pathway can actually produce glucose 6 phosphate, which is the second molecule in glycolysis. And of course, glucose 6 phosphate can be used for gluconeogenesis. It can help generate our final product of glucose, or it can be shuttled back and be used to break down through glycolysis. So we can actually build glucose 6 phosphate up through gluconeogenesis via the reverse of this pathway. This pathway, I want to point out, occurs in the cytoplasm. Again, this intertalk between pathways is important. So remember that most everything we've talked about, glycolysis, gluconeogenesis, pentose phosphate pathway, has taken place in the cytoplasm, except for the first step of our gluconeogenesis. Remember, that happens in mitochondria and that has to be shuttled out into the cytoplasm. So why do the pentose phosphate pathway? The normal pathway, taking glucose 6 phosphate and making ribose 5 phosphate, again, is generating NADPH and ribose 5 phosphate. NADPH is a very important molecule for oxidative stress. NADPH is an electron carrier, and it is going to help to take another molecule, glutathione, and reduce it. So by making NADPH, one of its main roles is to then be reconverted back into NADP. But by doing this, it takes an oxidized glutathione and then reduces it into a reduced glutathione. Glutathione is related to oxidative damage, and glutathione gets oxidized in the process of helping to reduce reactive oxygen species. So NADPH, by regenerating that glutathione, that reduced glutathione, is doing its job to help reduce overall oxidative damage to the cell. This pathway in general is one of the main producers of NADPH, and so it is extremely important for that aspect. The aspect of oxidative stress, we will rely on this pentose phosphate pathway. Another big reason to make NADPH is its involvement in fatty acids. So taking precursors and actually building up fatty acids through biosynthesis occurs by using NADPH. So if we want to make fats, when we start talking about anabolic pathways of actually building fats, NADPH is used to help build fats in our body. So it has this dual role. It helps in building fatty acids and building lipids inside of our body, and it helps with maintaining levels of glutathione to combat oxidative damage. Of course, the other product, ribose 5-phosphate, as mentioned, is used in DNA and RNA synthesis, or it actually helps in the synthesis of coenzymes, right? ATP and GTP and all of these nucleotide-based precursors, these coenzymes, need ribose 5-phosphate as part of their building block. Remember, it's adenosine triphosphate, so that adenosine molecule has a ribose sugar on it. Now, cells that aren't using this ribose 5-phosphate to make nucleotides and use in coenzymes, etc., can then take that ribose 5-phosphate and transfer it back into glucose 6-phosphate through gluconeogenesis, essentially building back up our sugars, but still allowing for the production of NADPH. Pentose 5-phosphate pathway, the PPP pathway, has two separate phases in which it can conduct this. So unlike gluconeogenesis, when we're rebuilding back up sugar, it's actually going to go shuttle through a different pathway. So we're not going to lose out on our NADPH. So breaking down glucose 6-phosphate through this pathway and then building it back up separately, we still can generate NADPH. So the reason that we would want to do this pathway is to make that NADPH to help with oxidative stress or to help with building up lipids. That's when this pathway is turned on. Or if you just have an excess amount of ribose 5-phosphate hanging around, then you might as well take it and build up glucose if you need energy. So let's talk about the oxidative phase of the pentose 5-phosphate pathway, the phase in which we're breaking down glucose 6-phosphate. You can see it happens in a couple of steps that are going to generate our NADPH, and also along the way, CO2. We're coming from a 6-carbon long molecule, and we're generating in the end a 5-carbon long molecule. So the carbon itself is released through a CO2. Now, once we generate our ribulose 5-phosphate during this process, ribulose 5-phosphate can then be used to generate ribose 5-phosphate for DNA or RNA. At this point of the pathway, you have a choice. Once you get to the ribulose 5-phosphate, you can say, okay, I need ribose 5-phosphate for DNA and RNA. This step is reversible. You can go back and forth between this ribose 5-phosphate back to the ribulose. Or ribulose 5-phosphate can be used to generate another molecule, which is xylose 5-phosphate. So again, this is just a rearrangement of the molecule, and it is reversible. if you're generating xylulose 5-phosphate, then you've committed to what is called the non-oxidative phase. That's the phase in which we are then going backwards to build back up glucose 6-phosphate. So again, once we get to our ribulose 5-phosphate in this pathway, we can decide I want to make ribose 5-phosphate and use that for DNA and RNA, or I can take my ribulose and my xylulose. I can convert between either, and I'm going to use these for non-oxidative PPP for building back up glucose 6-phosphate. So let's see what that looks like. Now, remember in the reverse, we're actually generating a 6-carbon molecule from something that has 5 carbons. So we have to generate glucose 6-phosphate from ribose 5-phosphate and xylulose 5-phosphate. This ribose or ribulose, right? So we're technically using ribulose and xylulose. Now, to do this, we're going to utilize a set of enzymes that are unique to this pathway. They're called the transketolases and transaldolases. These are specific to this non-oxidative phase of the pathway. But then our end products are going to feed back into our normal gluconeogenesis. So this is how it connects. Fructose 6-phosphate is going to be generated, and glyceraldehyde 3-phosphate is going to be generated, and these all can feed directly into the gluconeogenesis to then make glucose 6-phosphate. Here's a closer view of what that looks like. Remember, here is our oxidative phase. This is a breakdown of glucose 6-phosphate to make ribulose 5-phosphate. And during this process, remember, we generate some nice NADPH, 2 NADPHs, and a CO2. So 2 NADPHs and 1 CO2 are generated during the oxidative phase. The non-oxidative phase can then occur to cycle back into our gluconeogenesis or into glycolysis, in fact, if you wanted to use these molecules in any direction. Remember, gluconeogenesis and glycolysis are reversible. So depending on what the body's needs are, we're of course going to use these where we see fit. So ribulose, as noted, can be transformed into xylulose or ribose 5-phosphate. Both are which are going to be used during this process. One ribulose will form either one of these. So if we start with one ribulose and make one ribose, this actually needs a xylulose, so we have to use a second ribulose, and they combine to make these 2 molecules. This is a 3-carbon molecule and a 7-carbon molecule, which then combine through our series of ketolases and aldolases to make 1 fructose 6-phosphate and 1 erythrose 4-phosphate. So congrats, we made 1 fructose 6-phosphate. This can go directly into our glycolysis gluconeogenesis cycle. Now notice to do this, we had to use a xylose and a ribose, that was 2 molecules, just to make 1 fructose 6-phosphate. And we also made this erythrose 4-phosphate. Erythrose 4-phosphate can then further be made into a glyceraldehyde 3-phosphate, which can go into glycolysis, yay, and 1 fructose 6-phosphate, which also can go into glycolysis. But the way that it does this is by utilizing 1 more xylulose 5-phosphate. So in total, we had to use 2 xylose 5-phosphates and 1 ribose 5-phosphate to end up producing a fructose 6-phosphate, a glyceraldehyde 3-phosphate, and another fructose 6-phosphate. So how does that track? Well, that is 3 5-carbon long molecules, xylose 5-phosphate and ribose 5-phosphate. These are both 5-carbon long, which equals 15 carbons. And we generate from all of that interconversion a 6, a 6, and a 4, which all add up to, again, 15. So these can all feed into glycolysis. Now, the math for this is a little bit more complicated because although we are going to have to use these 3 molecules to generate 3 molecules, 3 full molecules, remember that glyceraldehyde 3-phosphate, it's only 1 3-carbon long molecule. And in fact, if we wanted to utilize it to build up glucose, then we would actually need 2 of them, right? We need 2 glyceraldehyde 3-phosphates to fully go into reverse. So to fully utilize all of our molecules to make glucose again, to make glucose 6-phosphate, we actually have to do that whole process I just talked about twice. So that means using 3 more molecules, a total of 6 5-carbon long molecules. That's 4 xyloses and 2 riboses. We're going to double everything here. And you can see now we made 2 fructose 6-phosphates. Those are good. They can go directly in. Another 2 fructose 6-phosphates. Again, they're good. They can go directly in. And now we have 2 glyceraldehyde 3-phosphates. They're no longer lonely anymore. They can combine with each other and go backwards through gluconeogenesis. So in total, when talking about this process, as I just detailed, we usually think of this non-oxidative phase as taking 6 5-carbon long molecules to make 5 6-carbon long molecules, ultimately. So again, we're going to take 4 xyloses and 2 riboses in total. That's 6 5-carbon long. They're both 5-carbon long. And we're going to make 2 fructoses here, 2 fructoses here. That's 4. And 2 glyceraldehyde 3-phosphates. These are only 3-carbon long, but they will recombine to make 1 glucose. So 2, 2, and then ultimately 1 more glucose is 5. Here's a simplified breakdown of that. Again, when we combine our first set of 5-carbon molecules, we end up getting 1 6-carbon molecule here, another 6-carbon molecule here, and we get this 3-carbon molecule, which needs, as you can see, this other half of the process to make our last 6-carbon here. So overall, this is where we're getting our 6 5-carbon molecules to ultimately generate 5 6-carbon molecules to fully do a non-oxidative phase of the pentose 5-phosphate pathway. Now, this results ultimately in making, rebuilding up 6-carbon glucose 6-phosphate utilizing part of gluconeogenesis. Remember, we're actually feeding these molecules back into gluconeogenesis or glycolysis. So the only parts of this that are unique to the pentose 5-phosphate are those transaldolases and transketolases that are rearranging molecules. It does not use ATP, which is nice, although when you feed it back into gluconeogenesis, remember, steps of gluconeogenesis will use ATP. So part of generating that glucose 6-phosphate potentially uses ATP. But these steps here do not utilize ATP. And it is a complete conversion of ribose 5-phosphate to glucose 6-phosphate, ultimately generating glucose 6-phosphate if needed. Again, the reason for doing pentose 5-phosphate pathway is if you are low on NADPH. It's a long, arduous cycle to take glucose 6-phosphate, break it down into ribose 5-phosphate, then use those ribose 5-phosphates to then rebuild up glucose 6-phosphate. But the reason for doing this cycle is to generate that NADPH, which is useful in other processes, right? So NADPH is actually the signaling molecule that will turn on or off this pentose 5-phosphate. Glucose 6 dehydrogenase, the molecule that is rate controlling of turning our glucose 6-phosphate or breaking it down, is the controlling enzyme here. And it is going to be stimulated by NADPH and inhibited very strongly by NADPH. So NADP plus, will stimulate it. NADPH will strongly break it down. So glucose 6-phosphate can either go through glycolysis or backwards through gluconeogenesis, that whole pathway involving ATP, or it can be shuttled through this pentose 5-phosphate pathway. And if we have an abundance of NADPH around, then we will not shuttle it through that pentose 5-phosphate pathway. It will not utilize that glucose 6-phosphate dehydrogenase, and it will instead go through glycolysis, etc. So this enzyme, this glucose 6-phosphate dehydrogenate, the first step of the pentose phosphate pathway, is an important enzyme in controlling the whole process of pentose phosphate. What if you have a deficiency in that enzyme? So again, glucose 6-phosphate is the first step of the oxidative phase of pentose 5-phosphate pathway. It is the rate controlling enzyme. So it is the most important enzyme in controlling all of pentose 5-phosphate, and if we utilize it. There are some inborn errors where there is known deficiency in this particular enzyme. It actually will predispose the infants to the breakdown of their red blood cells. You also see, following certain triggers, symptoms such as yellow skin, dark urine, shortness of breath, and feeling tired. And in general, these people are at risk for anemia. So remember, pentose 5-phosphate pathway is important in red blood cells. We see it utilized in red blood cells often. And this is why when we have an error with that enzyme, red blood cells in particular are affected. The reason they are so affected is because the reliance on the pentose 5-phosphate pathway to help counteract oxidative stress. If we cannot turn on the pentose 5 pathway, We're not generating that needed NADPH, which is used to help recycle glutathione. As you can see, here's our enzyme, glucose 6-phosphate dehydrogenase, first step of the PPP. It helps regenerate NADPH, which takes our oxidized glutathione to make reduced glutathione. And as you can see, this is going to combat things like hydrogen peroxide buildup, which is coming from any type of radicalization from oxidative stress in the mitochondria, ionizing radiation from UV, certain drugs, certain herbicides that you could ingest, um, certain foods in general. So if you have an error where you're not producing enough glucose 6-phosphate dehydrogenase, you thus are not helping recycle this glutathione and helping limit oxidative stress. So people who have this error, it can be fatal where they have cases of high oxidative stress on their body. So usually they're triggered by ingesting something, certain drugs, certain foods, herbicides, any of these things can trigger oxidative stress, high oxidative stress. They won't be able to combat it because they are not producing enough of glucose 6-phosphate and making their NADPH that they need. And thus they can cause lysis of their blood cells, hemolytic severity, hemolysis, etc. Now, the reason that this error seems to be passed on in people is because it does confer some resistance to malaria, which is related to the red blood cells. So having high oxidative stress in the red blood cells actually does allow some resistance to malaria, which is why this inborn error seems to track in certain populations where malaria is or was a large problem. Okay, so the summary for all of chapter 14. We started with talking about glycolysis. This is, of course, the process in which we break down glucose, our catabolic process, and fermentation, a process connected to glycolysis, which can continue glycolysis by energy extraction during anaerobic conditions. So fermentation allows us to continue doing glycolysis over and over again during these low oxygen conditions or in organisms that only function in low or no oxygen conditions. We then talked about gluconeogenesis, the process in which you can use a variety of metabolites like amino acids, et cetera, to funnel into making pyruvate, which can then be backwards synthesized into glucose. We looked at some of the differences between glycolysis and gluconeogenesis, those enzymes that are different, how they're differently regulated so that we aren't just futilely going through glycolysis and then going back up through gluconeogenesis, and how they're both made favorable. Then we also talked about the pentose phosphate pathway. This is a process in which cells can generate pentose phosphates and they can generate NADPH. And we talked about how those pentose phosphates, if they're not being used, can then be regenerated into glucose 6-phosphate without using ATP. So this whole side shunt process in which glucose can be regenerated and utilized to make NADPH.